mRNA occurring later in infection than the point that was tested. At the bottom of the gel, we observe a band representing the probe hybridizing to the 3' exon, which is the common product we mentioned before. At this point, the authors wanted to know how these results would change if complementarity was perturbed between the 5' splice sites of 12S and 13S and U1 snRNA. In order to do this, they developed different cell lines containing mutations at these sites. The first mutant cell line was co-transfected with the HR440 double mutation, which changed a GG sequence to AU in the 12S 5' splice site of E1A. It is worth noting that although the total number of base pairs between the mutant mRNA and wild-type U1 were predicted to be the same, the number of contiguous or uninterrupted base pairs would be disrupted. Lane 4 contains the results from this 12S double mutation in E1A paired with wild-type U1. We observe that there's no longer a band for the 12S splice product. Additionally, we can see that the band for the 9S splice product appears, and that there's more splice product for the 13S site. These results suggest that the base pairing between U1 and the pre-mRNA was impaired, leading to the inability to splice at this location. From this, we can conclude that base pairing at this location is necessary for splicing to occur. The mutant cell line was co-transfected with a mutation of both the 12S double mutation and a new mutation called U1-4U, which introduced a new mutation in U1 that restored contiguous base pairing with the mutant 12S splice site we saw previously. Lane 5 shows our results. Mutant U1 was predicted to have more complementarity with the 12S double mutation than wild-type U1, and we see that the results strongly resemble the wild-type trial. This would suggest that base pairing at this location is sufficient for splicing to occur. So, what can we conclude here? If we make a mutation in the pre-mRNA that cannot base pair with U1, we wipe out splicing at this location. If we make a compensatory change in U1 to restore base pairing, we can suppress this phenotype and splicing is restored. Thus, for this splice site, base pairing between the 5' splice site and U1 is sufficient for splicing to occur. The third mutant cell line contained the PM1114 mutation, which changed an A to a U in the 13S splice site. Similar to what we observed with mutating the 12S splice site, this mutation was predicted to disrupt continuous base pairing with U1. In Lane 6, we observed that the 13S single mutation had a similar effect to 12S double mutation in that it eliminated the 13S splice product. Once again, we can see that the concentration of the other splice product seems to increase. This supports our previous conclusion that base pairing is necessary for splicing in this location to occur. The last mutant cell line was co-transfected with both the 13S single mutation and a mutation called U1-6A, which introduced a new mutation in U1 that was predicted to restore contiguous base pairing with the mutant 13S splice site. Lane 7 shows the results. While we would predict from what we saw previously that the 13S splice product would be restored, this was not the case. So, what can we conclude from this? If we make a mutation in the pre-mRNA that cannot base pair with U1, we wipe out splicing at this location. If we make a compensatory change in U1 to restore base pairing, however, we do not suppress the phenotype and do not restore splicing. Thus, for this splice site, base pairing between the 5' splice site and U1 is not sufficient for splicing to occur. It was found after the 1986 experiment that the reason that the 13S single mutation and the U1 mutant did not restore the 13S splice product was because the mutation in the 13S single mutation eliminated complementarity with U6, another important SNRP in the splicing process. So what can we take away from all this? The initial purpose of the study was to find direct evidence for the proposed mechanism that the 5' end of U1 snRNA recognizes the 5' splice site in pre-mRNAs. We observed that while restoring base pairing between U1 and the 12S splice site following mutation allowed for splicing to occur, this did not occur when the same methods were applied to the 13S splice site. Thus, we can conclude that base pairing between U1 and pre-mRNA occurs, but restoring base pairing to the 13S splice site mutant is not sufficient for splicing to occur. Later experiments show us that binding between U6 and the pre-mRNA was perturbed, which is what impaired 13S splicing. Thank you guys so much for watching our video. Good luck studying and go blue!
Hello, my name is Andy Fan. Today we'll be discussing a 1991 paper by Lee and Goldfarb. They were investigating the exact repression mechanism of the lac operon by lac-I. For those who have access to it, we'll be looking at figures 6.8 and 7.8 from the 5th edition of Molecular Biology by Robert Weaver. This video was made for MCDB 427, the Molecular Biology class at the University of Michigan. Let's begin. Before we can discuss the experiment, let us first establish the general mechanism for transcription as background. Transcription is the process that takes the genetic information stored in DNA and produces an RNA copy of it. This process utilizes an enzyme, RNA polymerase, to accomplish this task. Transcription can be broken down into 3 parts: initiation, elongation, and termination. In this video, we'll focus mainly on the initiation component of transcription. Here's a diagram that presents the general overview of the 4 steps of initiation, so let's go into further detail of each individual step. We begin with the gene and the enzyme RNA polymerase. As we can see, the enzyme has a component attached to it called the sigma factor, here in blue. This factor is responsible for identifying specific sequences called promoters in the gene, so that it can begin transcription. As such, we consider sigma to be the specificity factor as it directs RNA polymerase to start transcription. As a result of this guidance, RNA polymerase is able to bind loosely to the DNA and produce a closed promoter complex. In order to proceed to the next step, this closed promoter complex needs to be converted into the open variant of the promoter complex. Here's a figure that demonstrates how this complex conversion process occurs. The closed component is derived from the fact that the double-stranded DNA is closed off from direct binding with RNA polymerase. In order for the complex to be converted into the open form, the sigma factor stimulates the core enzyme to melt these 2 DNA strands at the promoter and expose the DNA template strands of RNA polymerase. With the strands no longer closed off, the RNA polymerase can begin with the initiation process of transcription. Now back to initiation. RNA polymerase begins to incorporate the first few nucleotides the components of the LAC operon that the paper was interested in was the LAC gene. It is responsible for coding up for a repressor monomer that binds with itself to form a tetramer product. This tetramer binds to the operator LAC-O and prevents transcription of the downstream genes. If RNA polymerase is bound to LAC-P, the promoter region, as seen here, it cannot continue on transcribing due to this blockage. Now that we have addressed the background, we can now return to the topic of the experiment. The researchers Lee and Goldfarb were curious about the exact mechanism of repression for the LAC operon. I will now hand it off to Thomas to discuss these details.
The question that we're trying to answer right now is, how does the LAC repressor actually do its job? So here we've got a model of the LAC operon. We have our promoter here in green, the operator in red, and the LAC Z gene and so on here in this sort of tan-brown color. We know that transcription will occur when RNA polymerase is able to come here and bind to the promoter, forming an open complex, and then proceed with transcription. Our question is, what happens when the LAC I, this repressor, comes in and binds the operator? Is it the case that the repressor blocks the binding of RNA polymerase, in which case you just see that RNA polymerase is unable to even bind to the promoter? Or does the repressor block transcription elongation, in which case RNA polymerase would be able to bind to the promoter, it just would not be able to actually create a transcript? So now we're going to talk about the actual experiments done by Lee and Goldfarb in 1991 that investigated whether or not the repressor blocked RNA polymerase from binding to the promoter, or whether it simply blocked elongation. To investigate this, they performed a runoff transcription assay, which basically has as its goal to assess the transcription levels of a particular gene of interest, or, more importantly for us, to assess the potency of a promoter, here, the LAC operon promoter, so LAC promoter. So how does this assay work? Well, the first thing you need to do is you need to actually isolate your promoter and your gene of interest. So if we have here the E. coli genome, what we can do is we can make two cuts, one just a little bit upstream of the promoter here, and one in the middle of the Z gene. What this gives us is it gives us a small 123 base pair long fragment of DNA that consists of our control elements here, control, as well as a fragment of the LAC Z gene here. What we can now do is, in a test tube, we can add in the repressor for 10 minutes. We let the repressor incubate for 10 minutes, and at the end of the 10 minutes, we're going to see the repressor bind very stably, very strongly here, to the operator. Next, we can add into this test tube RNA polymerase, and this is where things get really interesting, because we're going to have RNA polymerase floating around in solution. And the question is, what happens here at the promoter? The first possibility is that RNA polymerase does not bind, so does not bind. In this situation, RNA polymerase is going to be floating around in solution, but the repressor will block binding. What we can do next is we can add in heparin, and heparin is an anionic polysaccharide, so that basically means that it has a bunch of negative charges on it. Now, RNA polymerase is actually a cationic protein, which means it has positive charges, so all the RNA polymerase that is floating around in solution will be scavenged, bound to, by heparin, which will block it from initiating transcription and thus binding to DNA. So all the RNA polymerase that is not bound to DNA already will be bound to heparin. Next, we can add in two things. We can add in NTPs, nucleotide triphosphates, and our inducer, IPTG. So, importantly, one of these NTPs has to be radiolabeled, so we can actually visualize what we're doing. We're going to radiolabel CTP. And the inducer, IPTG, its main job is to come here, bind to the repressor, cause a conformational shift, which releases the repressor from the operator. So the inducer makes sure that the repressor leaves the operator. So now we just got this blank piece of DNA, which is great, except that because we don't have any RNA polymerase bound to the promoter, you actually don't get any transcription of this gene, which means that at the end of this process, we get no radiolabeled runoff transcripts. The other possibility is that RNA polymerase does bind to the promoter, and indeed, it forms an open complex. In this situation, we still have RNA polymerase floating around out here, but some of it will have bound to the promoter. Now when we added heparin, we will block all of these free-floating RNA polymerases from binding, but you can't actually rip away this RNA polymerase that is already bound because it is formed an open complex. And open complexes are very stable. It's very difficult to rip this away once it's actually melted the DNA. Now when we added NTPs and our IPTG, you had the removal of the repressor, and now RNA polymerase in an open complex is all set to transcribe our gene. At the end of this process, we will see the production of radiolabeled transcripts, here with the C being a radiolabeled CTP. So what did the authors actually find? So, to recap, the authors wanted to figure out whether or not RNA polymerase was, in fact, able to bind to the LAC promoter, even in the presence of repressor. As a quick aside, you'll see that in the figure it mentions LAC-R here. LAC-R is the name for the repressor protein that's coded for by the LAC-I gene. So when you see LAC-I and LAC-R, we can interpret them both as repressor. So, to answer this question, the authors performed the experiment we just described, and they performed it in three different ways, as is evident by these three different lanes in this figure. The first lane corresponds to an experiment where they took the LAC promoter, they added an RNA polymerase immediately without any repressor present, after which they added heparin to scavenge any free-floating RNA polymerase, and then your radiolabeled NTPs. When they ran this gel, they were able to visualize the radiolabeled RNA transcripts, and they found that there were these two large, heavy bands up here at the top of the gel. These correspond to large runoff transcripts. So this indicates that RNA polymerase was able to bind, and then it was able to do transcription elongation, resulting in a large RNA molecule. We also see down here at the bottom these smaller bands. These correspond to those abortive transcripts we mentioned earlier. Recall that abortive transcripts are a normal part of transcription initiation. They imply that RNA polymerase was able to bind and form an open complex, and then proceed to create these small 7-nucleotide-long RNA fragments. In lane 2, the authors did the same thing, except this time what they did is they added in the LAC-I repressor before adding in RNA polymerase. So in lane 2, we see that the repressor has bound, and they found that there were no significant runoff transcripts formed, which is to be expected because we've added in the repressor, and we have not added in the inducer, right? There's no IPTG down here. So there's nothing to remove the repressor unless we get no runoff transcripts. However, intriguingly, we do see the formation here of these abortive transcripts once again. So this indicates that RNA polymerase was able to bind, form an open complex with the promoter, and create these abortive transcripts, but it wasn't actually able to get past the repressor and form a full runoff transcript. In lane 3, the authors performed the same experiment as in lane 2, except this time they did add in the inducer and the experiment. So when they added in the LAC-I, the repressor here, it bound to the operator and blocked RNA polymerase from actually transcribing, from elongating the transcript. But RNA polymerase was in fact still able to bind, as is evidenced by these abortive transcripts down here. Then when they added in NTPs and the inducer, this inducer alleviated the repressor. So the repressor left, and that RNA polymerase that had bound was in fact able to proceed with transcription elongation. So the potential mechanism of repression at this point seems to be that when we have no repressor, we form these abortive transcripts, and RNA polymerase is actually going to be able to transcribe the full gene, and we get mRNA. But when you see the repressor being bound, we see still abortive transcripts being made, indicating that RNA polymerase binds, forms an open complex, but it's just unable to get past this repressor. It's unable to elongate. So to summarize what we talked about in this presentation, RNA polymerase is able to bind to and form an open complex with the promoter like operon, even in the presence of repressor. The repression that we see may be due in fact to the repressor blocking transcription elongation, as we still see the creation of small abortive transcripts, indicating that initiation does in fact occur. Once we add in our inducer, either IPG or allolactose, then the repression is lifted, which allows any already bound RNA polymerase to proceed with elongation. However, this study was not without major limitations, the most significant of which was that the conditions used did not mimic physiological conditions, namely they were in vitro, and the amount of protein used was too substantial. Future studies did demonstrate that in physiological conditions, RNA polymerase is unable to bind to the promoter in the presence of repressor. Thank you very much for watching, and go blue!
Hello, this is Ari and Caroline from University of Michigan's MCDB 427 molecular biology class, and we are going to be discussing the GLIS cassette.
The GLIS cassette is a common molecular biology tool used to quantify transcription in vitro. Things that affect transcription levels are easily studied using a GLIS cassette, such as promoters, promoter elements, transcription factors, and activators.
You might be wondering, hey, why is it called a GLIS cassette? Well, in this assay, a long stretch of nucleotides that make up the GLIS cassette lack guanine, hence the G. As you can see on our image, the DNA in this plasmid is double-stranded. On the top strand, there is no guanine in the long stretch of nucleotides. Therefore, the bottom strand lacks cytosine, the C nucleotide you may be familiar with. We will refer to the top strand, the one lacking Gs, as the non-template strand, and the bottom strand as the template strand.
You may recall that in transcription, RNA polymerase reads the template strand and synthesizes a nucleic acid strand that is complementary to the template strand. Remember that RNA polymerase incorporates ribonucleotides into its RNA product, not deoxyribonucleotides, which make up DNA. The product of transcription, the mRNA transcript, therefore has a similar sequence to the non-template strand, but instead of thymine, RNA has uracil. While it is common to use a G-list cassette, you can perform a similar experiment using a C-list cassette or the less common A or T-list cassette.
In this assay, we rely on simple biological mechanisms to give very useful and reliable information. I will lay out an overview of how it works. First, we incorporate a promoter into a pre-constructed plasmid. This promoter will be located just upstream relative to the GLIS cassette, a stretch of nucleotide lacking guanine. Now, let's set up the GLIS cassette reaction so you can understand how it measures the amount of transcription. Once the promoter is inserted into the plasmid, we add RNA polymerase. This is an in vitro reaction, so we can control what ribonucleotides RNA polymerase can use for transcription. We just supply RNA polymerase with CTP, ATP, and alpha-32P labeled UTP, but no GTP.
We allow for transcription to occur. As RNA polymerase reads the template strand and incorporates complementary ribonucleotides into the growing transcript. For example, a stretch of DNA on the template strand reading TAG would result in a transcript reading AUC. Since A is complementary to T, U is complementary to A, only relevant to RNA as we have here, and C is complementary to G. Should RNA polymerase run into a C on the template strand, it would attempt to incorporate the G into the growing transcript. However, by not supplying any GTP, it would prevent elongation of the transcript. The RNA polymerase is therefore unable to continue and the transcript is released.
We then take the RNA products, load them on a gel, and do electrophoresis. Next, we do autoradiography or phosphorimaging, and we can see how much radioactive mRNA was made. On the audioradiogram, we can visualize and quantify the amount of transcription products. We could have set up this experiment using different promoters, the same promoter but mutated, or the same promoter with an activator present, to see how these changes affect transcription.
This is what a GLIS cassette looks like when it is used on a plasmid. In the plasmid show, there's a GLIS cassette sequence followed by DNA downstream that has a G on its non-template strand. Upstream of the GLIS cassette is the promoter. Please note that on the bottom graphic, the top strand is the template strand, written 3' to 5', which is unlike most other images you've seen of DNA. This still represents a GLIS cassette.
Now that you know how a GLIS cassette can be used and why its information is valuable, let's take a look at an example that was actually done. The GLIS cassette assay was used to observe mediator's effect on transcription. Mediator is a protein complex that is necessary for optimal activation of transcription in vivo. This next experiment we will show you was a real experiment conducted using a GLIS cassette to show mediator's effect on transcription activity with an activator present. Scientists inserted a CYCI promoter and a GAL4 binding site just upstream of the GLIS region. GAL4, a known activator, binds the GAL4 binding site and increases transcription by recruiting transcription machinery that make up the pre-initiation complex. In this specific experiment, they used a GAL4 BP16 activator hybrid. This hybrid has the DNA binding domain of GAL4 and the activating domain of BP16. From now on, we will refer to this hybrid protein as activator. Scientists added different amounts of mediator, measured in micrograms, to test its effect on the amount of transcription.
Let's break this gel down lane by lane. We can see that in lane 1, there was no activator nor mediator present in the transcription reaction. A faint band on the gel, which is difficult to see, indicates that a basal level of transcription occurred. Looking at lane 2, we can see a much darker band, which indicated that more RNA products were made in this reaction. Lane 2 shows that addition of the activator increased transcription. As we look
look at lanes 7 through 10, more RNA transcripts are being made. In those lanes, mediator and the activator were both present in the reaction. Looking at lanes 7 to 10, we can see that the concentration of mediator used in the reaction is increasing. Since the bands got darker from lanes 7 through 10, this indicates that more RNA transcripts were made as more mediator was added in the presence of the activator. To test to see if mediator could increase transcription without an activator present, they performed the same reaction as in lanes 7 through 10, but without the activator. In lanes 3 through 6, we can see only faint bands, which indicate that there is very little transcription occurring. This led scientists to believe that mediator only increases transcription when there's an activator present. With this in mind, the gel bands in lanes 3 through 6 should appear identical since a mediator does not affect transcription amounts when no activator is present.
This graph correlates to a replicate experiment and is another way to represent the amount of transcription that occurred with activator and without activator. The band's radioactivity was quantified, and the points on the lines indicate how much transcription occurred. The x-axis is the concentration of mediator added, and the y-axis represents the amount of transcription. You can see that when no activator is present, there is a constant amount of transcription as the concentration of mediator increases. This line is blue and is labeled basal on the graph. This shows that while transcription can occur at a low level without activators added, the mediator does not affect transcription when no activators are present. Hence, while lanes 3 through 6 on the gel should have had identical bands. When the activator is present and scientists increase the concentration of mediator, there is an increasing amount of transcription, as shown by the almost linear rising green line labeled activated on the graph. These results show that mediator affects transcription when there is activator present. In conclusion, when an activator is not present, increasing the amount of mediator does not affect transcription. However, when an activator is present, increasing the concentration of mediator does increase the amount of transcription.
Make sure to remember these three things. 1. The Gillis cassette studies the transcriptional strength of promoters in vitro. 2. It's called the Gillis cassette because there are no G's in the non-template strands. And 3. This assay was used to study the effects of mediator on transcription. Thank you very much for watching, and go blue.
Hi, this is Katie Hong, student in MCD 427 Molecular Biology at University of Michigan. Today we're going to talk about an experiment done by Frank Grossfeld in 1987, which answered a big question at the time, which is, how does transcription occur when DNA is complex with nucleosomes? As we'll talk about later, DNA complex with nucleosomes cannot be transcribed, leading to the idea that there are parts of DNA that are nucleosome-free. This video corresponds to figures 13.19 and 13.20 from Molecular Biology by Robert Weaver. The structure of chromatin is important for transcription. DNA will coil around these blue proteins called histones, which forms nucleosomes. We call these heterochromatin. Due to this structure, RNA polymerase cannot access the DNA, and transcription does not occur. So this brings up the question, how can DNA be accessed by RNA polymerase when nucleosomes are present? We now know that actively expressed DNA has breaks in the histones, forming the less dense euchromatin, which can be accessed by RNA polymerase for transcription. The enzyme used in this experiment is deoxyribonuclease, also called DNase, which cleaves double-stranded DNA at the phosphodiester bonds in the backbone. Chromatin completely complex with nucleosomes will be protected against DNase. However, chromatin that is being actively transcribed has nucleosome-free regions and is considered to be DNA sensitive. These regions are called hypersensitive sites and tend to be control regions. Now that we have some background information, let's take a look at the experimental setup. First, we have this DNA that is completely complex with nucleosomes. It is inactive and not sensitive to DNase. This is just showing the region of the DNA. There's actually lots more. We treat the nuclei with only DNA, so it is not overcutting the DNA. We want one cut per region of DNA. Since this whole segment is protected by nucleosomes, the DNase does not degrade the DNA. We then remove the nucleosomes. This is an important step, as our next step is to treat the naked DNA with restriction enzymes and electrophoresis disabled aerosol. The restriction enzymes let us make our fragment a specific known size. In this case, they cut us in green arrows. If we don't remove the nucleosomes, the restriction enzymes won't cut, so we won't get that specific fragment size. In this case, we get a fragment that is 13 kb, which you can see right there. Now let's take a look at DNA that is not completely complex with nucleosomes. In this example, there's one spot without a nucleosome. We know this spot without a nucleosome is a hypersensitive spot for DNase. We then treat this DNA with only DNase. However, in this case, the DNase will cut at the hypersensitive spot marked by the red arrow. Then we remove the protein and treat with restriction enzymes. This leaves us with two fragments, 6 kb and 7 kb. We then gel electrophoresis. Since this is on the gel, we can visualize these bands at 13 kb on the left and 7 and 6 kb on the right. It is important to note there are other fragments in the experiment that will be on the gel. The DNA we're cutting is much larger than what is pictured here. To visualize the bands that will be unfolded, the 7 got a gel, so there are single-stranded DNA fragments on the filter. We've shown them here, but they're actually invisible. To visualize, we incubate with a hot probe that will hybridize to single-stranded DNA on the filter, specifically